Sure, and that's actually very related to what I just talked about regarding the PKMYT1. So here we have generated cell lines where we knocked out the endogenous cyclin A, and these are HMECs, human mammary epithelial cells, and then we inducibly express either the full length or the low forms of cyclin A. And under these conditions, we see that the mechanism by which the full length or the low form mediate genomic instability is very, very different, in the sense that with the full-length cyclin A, which only resides in the nucleus, when you overexpress it, the cells actually die because they undergo so much genomic instability they cannot repair. However, when you put in the low forms, because they are going back and forth from the nucleus and the cytoplasm, they actually are enriched in the cytoplasm, the cells have found a way to repair or to tolerate the damage. So they accumulate genomic instability, and they become, in fact, more tumorigenic under those conditions. So we went on and examined under the mechanism by which this genomic instability tolerance comes into play. What are some of the molecules that are involved? And this is through a number of different molecular analysis. And then based on that, we also then identify targets that could be used to treat these cells. And then for that, we actually went to the TNBC, and we identified a number of inhibitors that specifically target these pathways that are expressed or induced when the low forms of cyclin are induced. And in fact, this work has recently also been published in the Journal of Oncogene. So I think that also provides additional insight into our findings.